Oligonucleotide Discovery and Development, brought to you by Pharma Intelligence and sponsored by biotech. Today's session is focused on oligonucleotide drug discovery, and we'll have presentations from experts involved in providing state-of-the-art technical support to the discovery stages of an oligonucleotide therapeutic program. My name is Mari Gerard, Managing Editor for Custom Content at Pharma Intelligence, and I'll be your host today. Before we jump into the presentations, let's go over some quick housekeeping items. First, if at any time you are having technical difficulties, click on the Test Your Connection button, and this will take you to a connection checker and FAQs page. If you are experiencing any technical issues, there is a Chat Now function in order to go through to our support team who can assist you. If you'd like to enlarge the presentation, just click on the Maximize icon or drag the lower right-hand corner of the presentation window to enlarge. There will be a Q&A at the end of this presentation, so if you have questions, we really want to hear from you. To ask something to our presenters, simply type your question into the question window on the bottom of your screen and then hit submit. We'll be answering as many questions as possible during the Q&A, and if there are extras, we'll try to answer you after the webinar via email. Because we know you'll want to listen to this webinar again, today's session is also being recorded and will be available to view on demand in around 48 hours. I would now like to introduce our speakers for today's session. Full details of these are available to the right of your screen in the bio section. First, we'll have Mark Edbrook, an independent consultant in the oligonucleotide space. Then we will have Chong Zhou, Executive Director for Discovery Biology at biotech. Then we will have Jun Zhou, Senior Principal Scientist and Associate Director at biotech, followed by Nan Zhao, Principal Scientist 3, DMPK Service Department, also at biotech. And finally, we'll have Rob Goodnow, VP of Medicinal Chemistry at Stoke Therapeutics. And with that, I will hand things over. Mark, the floor is yours.
Hello and welcome everyone to this webinar about oligonucleotide drug discovery. My name is Mark Edbrook and I've been working in the field of therapeutic oligonucleotides for over 25 years. So I have a question for you, and that is, what is exciting about the drugs that are depicted on this slide? And the answer is that they are all oligonucleotide-based medicines that have been approved in the last 2 years. And this demonstrates the extraordinary progress that's been made by this drug modality in recent times. In fact, this now means that there are more than 16 oligo-based drugs that have been approved, and that's not including N of 1 drugs, for example, Millicen. The 16 include most forms of oligonucleotide drugs, including single-stranded, double-stranded drugs, different delivery routes of administration, types of formulation, and different types of diseases. So oligonucleotides can be used in different ways to affect gene expression. They can be used as gapmers to lead to mRNA degradation. They can be used as aptamers. They can be used to inhibit nuclear steric blocking and using splice switching or splice skipping oligonucleotides. They can be used in the blockage of assembly of RNA binding factors. They can be used in activation or, in fact, inactivation in the innate immunity. They can be used to mimic or antagonize microRNA activity. They can be used as agamers and block translational inhibition. And of course, there is RNA interference, where oligos can be used as small interfering RNAs or as short hairpin RNAs. And this is illustrated on this diagram from a review paper by Susan Hammond in EMBO Molecular Medicine last year. And so from left to right, we have illustrations of when oligos get inside cells and can act as gapmers, as aptamers, as antisense oligonucleotides, can be involved in TLR signaling, involved in microRNA agonism or antagonism, involved as agamers, or lastly, involved in RNA interference. And the reason why this extraordinary progress has been made is illustrated in this diagram, where the evolution of oligonucleotide chemistry and the better biological understanding of how oligos work has progressed tremendously over the recent times. And again, this is summarized very nicely in this figure from a review by Anastasia Karova and Jonathan Watts in Nature in 2017. So key aspects of what you're going to be covered during the two-part webinar that you're attending now will include oligonucleotide chemistry and design, biology, DMPK, toxicological assessment, and manufacture, purification, and formulation. So please enjoy the presentations and be ready with any questions for the Q&A at the end. And as Mari said, as you're going through or listening to the talks, you can put questions into the Q&A box at the bottom of the screen. Okay, thank you. And I'll now hand you over to the first speaker.
Hello, everyone. This is Chung Zhou from Discovery Biology, WuXi AppTech. I'm glad to have this opportunity to introduce our oligonucleotide biology platform. Oligonucleotide mainly included antisense oligo, siRNA, microRNA, aptamer. There are more than 10 oligo drugs marketed so far. Since antisense oligo and siRNA interact with the target RNAs with the complementary base pairing mechanism, it is convenient to use computer programs to design oligo sequence. Theoretically, all the RNAs can be targeted, including the traditional undruggable targets for the small molecule and antibody drugs. Therefore, the oligonucleotide is considered to be the third major therapeutic modality. The advances of antisense oligo and siRNA drugs mainly are attributed to the breakthrough of the chemical modification and the delivery methods. Chemical modification can increase the oligo stability, decrease immune toxicity, and off-target effect. The delivery tools can improve the organ targeting efficiency, especially GalNAc can specifically and efficiently target hepatocyte. Currently, there are a lot of researches to explore the actual hepatic delivery of oligos to treat the other disease. So similar to the discovery of the other type of drugs, for oligonucleotide drug discovery, we need to improve the potency and decrease the toxicity of the compound. Our service covers in vitro, in vivo activity evaluation, delivery efficiency, and early tox, including the immune tox and off-target effects and the others. The first step for the oligo drug discovery usually is sequence design. This is a flow chart of our siRNA design pipeline. We also have a commercial database can filter out the label or label the patented sequence. For antisense oligo, we are also building up the capabilities, and hopefully we can provide the service later this year. Next step is for the oligo drug discovery usually is to evaluate the compound activity in cell-based assays, in the transfection assays. So we have more than 400 cell lines in WuXi Biology Bank. Also, we can access to some patient-derived fibroblasts and some primary cells. So we can choose the optimal cells for the compound activity evaluation. For antisense oligo, there are two major mechanisms. One is to degrade the mRNA through the RNase H-dependent degradation mechanism. The other one is to regulate the splicing. So we may use some patient-derived cells for the splicing mechanism antisense oligo compound. For the mRNA degradation, ASO and the siRNA compound, we can detect the mRNA expression level by RT-PCR or the branch DNA. And for the splicing, we can use the capillary electrophoresis or Sanger sequencing and NGS sequencing to evaluate the intra-axonal inclusion and exclusion effect. And the next, we may need to evaluate the protein levels by different methods. And then for some immature target, really to do some cell-based function assays, we can provide the customer service for this part. And also, I want to mention that for some targets, the expression level is very low in the cell lines or the primary cells. We may need to use an alternative method, PCI check reporter. And in this reporter assay, the targeted mRNA is inserted after the stop codon of the reporter gene, and the reporter activity can represent the chimeric mRNA level. So for the targets with very low expression level in cell lines, the reporter is a very useful tool. And the assay is also lower cost compared with the RT-PCR or branch DNA method. Besides, the PCI check reporter is also useful for evaluation of the off-target effect. Currently, we have validated a lot of in vitro screening assays for more, up to now more than 50 targets. Here are some example data of the RT-PCR assay in a cell line by transfection of the compound. In the primary screening, usually we recommend to do 4 concentration or 3 concentration, and then select a compound with good activity to do those response curve EC50 determinations. And I mentioned PCI check reporter is an alternative method for the compound evaluation. Here, we also put some example data for the reporter system. You can see, in generally, the reporter system has the consistent data compared with the RT-PCR method. And the data variation and the reproducibility is acceptable. Since the oligos are relatively large and negative charged, so it is very critical to deliver the compound into cells. For the 5 marketed siRNAs, 1 was delivered by LNP, the other 4 was delivered by GalNAc conjugate technology. So it is very critical for oligo compound to find out an efficient delivery method. For example, use the GalNAc, LNP, PNP, lipid, or the peptide conjugation. We can provide a service to improve check the cellular delivery effect. Taking the GalNAc conjugation method as an example, we develop cell-based binding and uptake assays. GalNAc can bind to the SPGR receptor on the cell membrane and induce the receptor mediated endocytosis. We use a fluorescence labeled oligo compound and incubate the compound with the different type of cells. You can see that the HepD2 and the 293A cells, the GalNAc SPGR binding activity is very low. However, the fresh primary mouse hepatocyte and the cryopreserved primary human hepatocyte has much higher SPGR binding activity. So furthermore, we use the primary human hepatocyte to develop a competition method to evaluate the compound activity of the novel GalNAc compound. So for the other, next we may need to do some free uptake assays, which means we treat the compound with the cells without transfection and detect the target mRNA or protein levels. Then we can evaluate the combined efficiency of the oligo sequence and the delivery tools. So since in the cell lines, the GalNAc uptake activity is very low. So for this free uptake assay, we need to use the primary cells. Here we show some example in the primary human hepatocyte and the primary cyno monkey hepatocyte. And then also we can isolate some fresh mouse hepatocyte from normal mouse or transgenic mouse or pharmacology mouse models to some specific targets. So for the other delivery method, we can also set up method by using some optimal cell lines or primary cells. We also can do in vivo delivery efficiency evaluation for oligo compound, including such as the lab imaging system with the fluorescence labeled oligo in mouse for the biodistribution studies. And besides, we can also detect oligo in plasma or in tissues by different methods, including LC-MS. And my colleague Nan will talk about LC-MS in details later. And in our team, here I would like to talk about, we use the stem loop PCR to determine the risk associated siRNA. So we use the AGO2 antibody to pull down the risk complex, and the siRNA associated with the AGO2 complex, risk complex can be detected by the stem loop RT-PCR method. The left panel is the results of the cell line validation results, and the red panel is the results from animal studies. We detected the total siRNA concentration in liver and also the risk associated concentration. You can see that the kinetics of the two type of compound, the total concentration and the risk associated concentration is different. For the total siRNA, the concentration decreased since 4 hours continuously. For the risk associated siRNA, relatively it maintained stable from day 7 to the day 28. And in general, the kinetics of the risk associated siRNA is more relevant with the clinical data. In clinical, siRNA compound, the GalNAc siRNA compound can be treated every 3 months or half a year. And also we can see that the siRNA, risk associated siRNA concentration is much lower than the total value. So it is consistent with the literature report that the endosome release efficiency is very low. So to improve the endosome release efficiency is objective of to improve the compound potency. So this method for the risk PK evaluation method should be a very useful tool for the related researches. And next, I would like to introduce about our animal models for the oligo compound evaluation. Since antisense and ASO compound, the mechanism is used the complementary base pairing mechanism. So we need to consider the homology between the animals and the humans, especially for the compound. The similarity between the mouse and human is very big. We need to consider to use the humanized mouse model for the compound activity evaluation. First, for the liver target, we can use the hydrodynamic injection mouse model. And hydrodynamic injection is to inject the plasmid DNA with the human genes into the animals by the tailwind within 5 seconds. And then it can result in the plasmid DNA entering into the hepatocyte. However, the expression of the transgene is usually transient. So this model can be used for the liver targeting, short-term compound evaluation. And the second model is the AAV mouse model. AAV is a widely used gene delivery tool, and the transgene expression level is very stable, as long as many years in the clinical. And in our labs, also we observed it is stable more than 10 months. And by using different type of AAV, we can target the different organs. For example, AAV8 for liver, AAV PHP point B is for CNS. So we can use AAV to introduce the human gene in different organ of the mouse for different targeting oligos. And also the model is useful for the long-term compound evaluation. The third model is transgenic model. Usually we prefer to use lock-in model. And by using the transgenic model, not only we can detect the compound activity on the target gene, target protein, and biomarkers. Also, we can use the transgenic mice for the pharmacology model establishment. However, usually it takes a long time to get the animals. If for the liver targeting, we can also use liver humanized model, and the liver human ratio can be higher than 30 or 90%. The advantage of this model is it is useful for the on-target and also off-target, because all the hepatocytes are human genes. But the cost may be much higher than the other models. In each project, we may need to choose the suitable model, may combine different models at different stages. So we can discuss with the client, share our experience and opinions for each specific project. In this slide, I show some example data for the HDI mouse. The left figure is the lab imaging of the BABC mice hydrodynamic injection with the luciferase plasmid DNA. You can see that the luciferase activity decreased very quickly. So usually for the HDI model, we pre-dose the animals and then inject the plasmid DNA. And this is a transient model. However, the data have acceptable and good reproducibility. You can see the data. The second study, the 3 doses showed very good dose-dependent effect. This slide, we put some example data of transgenic mice and the AAV mouse model. I just want to mention that for transgenic mice, we need to confirm the sequence before we use it, especially, for example, confirm whether the UTR sequence is included. And for the AAV model, I want to see that we can provide the AAV generation in-house to support the model establishment. Except for the mouse models, also we can provide the large animal services. So we put some example data here. For monkey, we can use various administration methods, including normal IV, IC injection, and also some special injection, for example, IT, ICV, and so on. We can also do the liver biopsy to collect samples. And also we have very comprehensive post-lab analysis platform to support the PD study in both the monkey and the mouse studies. At last, I would like to briefly talk about our capability of the off-target and early tox studies. We can access the general cytotoxicity and also to evaluate the immune toxicity. Oligo compound can activate the toll-like receptors, RIG-I, M85 pathways. So we can use the reporter genes to evaluate the compound specific activity on these pathways. And also we can detect the downstream cytokines of these pathways, usually in human PBMCs, but also we can do some animal immune cells, for example, PBMC from monkeys. Moreover, for the sequence dependent off-target is a major cause for the toxicity. So we also have RNA-seq platforms can do some RNA-seq analysis to support the off-target evaluation. Usually we provide the standard analysis, data analysis, including differential gene expression profiles, and also we can do some sequence-based off-target evaluation. Besides the biology support for the oligo discovery, we can work with other WuXi BUs to provide integrated service. So my colleagues in the other BUs will introduce the other WuXi capabilities of the oligo discovery. So next, my colleague Jun Zhou from the chemistry team will talk about the chemistry part. Thank you for your attention.
To the webinar. My name is Jun Zhou, joining from WuXi AppTech. Today, I'm going to talk about oligonucleotide chemical modification and conjugation. Chemical synthesis of therapeutic oligonucleotides relies on solid phase synthesis using phosphoramidite chemistry. The synthesis typically starts from CpG solid support, onto which the phosphoramidite building blocks are sequentially coupled until a full-length oligo is formed. This process has been fully automated using a variety of oligosynthesizers. At WuXi Chemistry, our facility is equipped with two major platforms, including Mermaid system for small-scale and library synthesis, and Atkala system for gram-scale and large-scale synthesis. To enable further conjugation or labeling, a reaction handle can be attached to the 5' end. For example, a amino modifier that allows for further amide coupling on 5' terminal. Upon completion of a sequence assembly, the oligo is released from the solid support using ammonium hydroxide. The cooled products are purified by reverse phase HPLC or an ion exchange chromatography to provide the desired oligos with high purity. In some cases, post-synthetic oligonucleotide conjugation is performed in solution phase to link with a different molecule. For example, amide coupling to attach a peptide as an active ester to the 5' amino modifier. The final oligo product is analyzed by UPLC TOF mass for purity determination and mass identification. Are most widely used as antisense oligonucleotides or siRNA. Since unmodified RNA can easily undergo nuclease degradation, a wide range of modified nucleic acids have been incorporated into therapeutic oligonucleotides to improve the stability, to fine-tune the drug-like properties, and to enhance the delivery of oligonucleotides. For siRNA, in addition to 2' methoxy, 2' fluoro, the UNA, GNA, and 5-VP nucleotides are incorporated to the antisense strands. And for ASO, a broad range of breached nucleic acids are introduced to the flanking region of the gapmers. Conjugation to targeting ligands at 3' or 5' terminals provides the opportunity to deliver oligonucleotides to specific tissues and to increase the half-life and retention, as well as intracellular uptake. The targeting ligands could be a peptide or carbohydrate, for example, GalNAc, lipophilic moieties, PEG, or a small molecule. At WuXi AppTec, we provide a comprehensive collection of modification and conjugation, and all this can be pre-made at building blocks on shelf, ready to go for oligonucleotide and conjugation chemistries. GalNAc is of highest interest to conjugate with ASO or siRNA to deliver the oligonucleotide to liver hepatocytes while specific binding to ASGPR with high affinity. From a synthetic perspective, trigalNAc ligand can be attached to either 3' or 5' terminal of ASO or siRNA sense strands through a variety of conjugation chemistry. For 3' GalNAc conjugation, the primary option is to load the trigalNAc onto CpG solid support, which serve as a start point of oligoassembly. For 5' terminal conjugation, the trigalNAc can be introduced as a phosphoramidite on solid support or attached through amide coupling or click chemistry in solution phase. At WuXi Chemistry, we have a successful track record in delivering more than 3,000 GalNAc conjugated siRNA on a variety of scales and more than 30 novel GalNAc ligand synthesis. Lipid conjugation of oligonucleotide has proven to enhance nucleus stability, prolong half-life and retention, and to increase intracellular uptake. The lipid conjugation moieties include long-chain lipid acids and other lipophilic molecules such as cholesterol and tocopherol. These lipid moieties can be linked to 5' ends through phosphoramidite coupling on solid phase or post-synthetic conjugation, including amide coupling or click chemistry in solution phase. For 3' lipid conjugation, The lipid moiety can be preloaded to CpG solid support, which is similar to GalNAc conjugation. Peptide conjugation can enable cell penetration and cell targeting delivery of oligonucleotides to specific tissues. For example, cell penetrating peptides, also called CPP, that binds with negatively charged cell surface proteoglycans across the cellular membranes, increases cellular delivery of oligonucleotides. Cyclic IgD peptide is a tumor targeting ligand and thus can be used to deliver oligonucleotides to tumor cells for cancer therapy. Peptide can be conjugated to either 3' or 5' ends through a variety of conjugation chemistries, including click chemistry, thiol-maleimide conjugate addition, and amide coupling. Prior to conjugation, the peptide moiety can be modified by appropriate functional groups that enables the conjugation. At WuXi Chemistry, we have established a highly convergent and versatile dual conjugation platform wherein an extensive collection of reaction handles can be introduced to either 3' or 5' terminal of an oligonucleotide. And these reaction handles can enable a series of conjugation chemistry to link with a broad range of molecular modalities. Moreover, the selections of appropriate chemistries that are orthogonal to each other would allow for sequential conjugation with two different ligands at both ends. Take one example. An azide linker is installed onto the 3' end that allows for copper-free click chemistry to attach a library of BCN modified lipids. And on the other end, the reaction handle can be in a terminal alkyne for copper catalyzed click chemistry to introduce a fluorescent tag. Alternatively, the 5' end linker can also be an amino modifier that can react with the peptide carboxylic acid active ester or maleimide linker that would couple with the thiol-containing peptide. The dual conjugation strategy is highly convergent and enables rapid installation of various ligands at both ends. At WuXi Chemistry, we provide one-stop solutions to early-stage discovery of therapeutic oligonucleotides, and our services include synthesis of special phosphoramidite monomers, various oligonucleotide modalities, and the best solutions to oligonucleotide conjugation.
Nucleotide therapeutics. Firstly, I want to give a brief introduction about DMPK. The purpose of DMPK is to study the characteristic interaction of a drug and the body for its absorption, distribution, metabolism, and excretion. In other words, DMPK is to figure out how the compounds of interest get into the body and how they move within the body and how they get out of the body. It can help assess the safety and efficiency of a new therapy. The DMPK of a new therapy should be studied and evaluated throughout the whole process of drug development. It can significantly improve the successful rate of the translation of a preclinical result to clinical manifestation. Now let me go back to the oligonucleotides. Oligonucleotides are a unique class of compounds that share characteristics of both large and small molecule. They're like a large molecule drug because they have relatively high molecular weight and possess excellent target specificity. However, they also share many characteristics of small molecules, including synthetic production, relatively simple chemical structure, and inclusion of chemical modification to improve its performance. This identity brings its unique ADME characteristic. So oligonucleotides is usually administered by IV infusion and subcutaneous injection or direct injection. After absorption, oligo are distributed within minutes to hours and rapidly uptake into tissues, mainly in the kidney and liver. And their exposure is much higher than that in plasma. The half-life of oligonucleotides in my organs, like liver or kidney, is from weeks to months, but in plasma it only takes few hours. Oligonucleotides is mainly characterized by extranuclease and endonuclease, resulting in short MERS and eliminated through urine and feces. And then next, I want to share some information about how we studied the distribution and metabolites of oligonucleotide. Okay, here. The distribution refers to the movement of a drug between intravascular and extravascular compartments of the body. Because the protein binding and unbound forms of drugs can affect their distribution in the body. So it suggests to conduct in vitro protein binding study in early discovery. Ultrafiltration, ultracentrifuge, and electrophoric mobility shift assay are always used to determine the protein binding characteristic of oligonucleotides. For in vitro study, the biodistribution is requested not only for the oligonucleotides, but also for its delivery system, or the individual components of the delivery system, and the combined final product like the protein. The biodistribution data is always obtained by either analyze the main tissues separately or based on the radiolabeling or image for the whole body. And then it's about the metabolism. Different from small molecule, oligonucleotides is mainly catalyzed by extranuclease and endonuclease, resulting in short MERS. The short MERS may be pharmacologically active and interference with bioanalytical assays. Therefore, it's also important to investigate the in vitro and in vivo metabolites during the preclinical stage or discovery stage. Here is the workflow of metabolic stability assay. For the in vitro study, the sample was obtained by incubating oligonucleotides within liver isolate, hepatocyte, or liver homogenate from different species. While for the in vivo study, the sample was obtained from dosed animals. All the samples were extracted and analyzed by high-resolution mass spec, and then potential metabolite could be identified by using the software such as the Biopharm Finder. As a consequence, the in vitro and the in vivo metabolic pathway will be found. Okay. Here is summary the key in vitro and in vivo ADME studies of oligonucleotide in the preclinical stage. It shows that the quantitative analysis of oligonucleotides from different assay types involves a variety of biological metrics and the target analyte. The target analyte is not only the oligo of interest, but also the oligonucleotide that may found as metabolites. In addition, the propagate of pharmacodynamic biomarkers, such as target mRNA, target protein, or downstream biomarker that reflects the modulation of the target protein, should be also measured. Therefore, it's important to develop and validate a proper bioanalytical assays to support oligonucleotide quantitation in biological metrics. However, the bioanalysis of oligonucleotide faced many challenges, requiring high laboratory platform construction and technical personnel expertise. As shown in the previous talk, the oligonucleotides has unique physical chemical properties like its size, diverse chemical modification, and the polyanic nature. It requires advanced and unique analytical strategies. During the method development of bioanalysis, many factors need to be considered, such as the bridge between different platforms, multiple biological samples used for the PD and PK evaluation, and the interference from the impurity and metabolites that could interfere with the assay. Especially for the immunogenicity studies, the ligand binding assay always need to be optimized to avoid interference from the negative charge protein, the cross-reactivities of the anti-drug antibody to endogenous RNA and DNA. Many methods have been developed and validated for quantitation of oligonucleotides in vivo and in vitro ADME studies. The liquid chromatography mass spec and the ligand binding assays such as ELISA and qPCR are widely used for oligo quantitation. However, there are more and more techniques such as branch DNA, ddPCR, fluorescence illumination image, and bioillumination image applied in this area. They can be generally classified by 4 main categories and identify the oligo of interest by charge, mass, binding, or imaging. It's important to choose the correct IC, which can help to indicate by the individual molecular. The method selection should account for the chemical structure of the oligonucleotides, the requirement of the assay sensitivity, the sample type, and the throughput, and even the development stage of your study. Our DMPK department provides a robust bioanalysis solution to accelerate the development of oligonucleotides from the discovery to late clinical stage. So far, we have developed multiple reliable bioanalytical platforms with high sensitivity, selectivity, accuracy, precision, and throughput. There are LC mass spec, LC high resolution mass spec, LC fluorescence detection, and ligand binding IC, and qPCR. Those platforms can support the oligonucleotide studies while viral stage. For example, the LC-MS method, which is used for the small molecule, can help measure an oligo's concentration in target organs, such as the liver, kidney. This IC can achieve high specificity, accuracy, and precision. But as the program processes to later stage of preclinical and clinical studies, it needs more sensitive method to measure lower concentration in vivo samples. The ligand binding, such as the hybridization ELISA, MSD, or qPCR are very helpful in this area. However, for this method, we may need to design the special probe and screening and synthesis this probe before we start the real method development. Okay, from the regulatory perspective, there is still some ambiguity for oligonucleotides. Although the US FDA considered oligonucleotides as a drug, not a biologist, should follow a regulatory framework of small molecular drugs for new drug application. However, the immunogenicity data, which is required for a biologist, was provided as part of the new drug application filing. It makes us to think about is which way we need to follow. Recent draft guidance from the FDA addresses the IND submission of individualized antisense oligonucleotide drug product, and also the clinical pharmacology consideration for the development of oligonucleotide therapeutics. With the development of oligonucleotide therapeutics, we believe in the future, there will be more and more guidance released by the regulator for the oligonucleotides, and we will pay close attention to that. Okay. Our DMPK service department provides comprehensive in vitro and in vivo ADME solution from high throughput screening and in discovery through the drug development phases, including in vitro ADME, rodent PK, large animal PK, non-GLP bioanalysis, drug-drug interaction, metabolite profiling and identification, non-clinical mass balance, tissue distribution, and radiolabel human AME studies. Beyond DMPK, our skilled team and toxicologists provide safety assessment across a wide range of species and through various routes of administration. We also have a bioanalytical service team to provide and through various to support your drug discovery and GLP compliance. We worked closely to offer
in oligonucleotides. I'd like to tell you a little bit about how antisense oligonucleotides are applied in Stoke's Tango therapeutic platform. First, a disclaimer saying simply that I may own some stock here, and this is for educational purposes alone. What I'd like to talk about is what is Tango and how are antisense oligonucleotides useful for that approach? What are some of the design considerations for antisense oligonucleotides, and what are the tools that we have to work with? We've heard already of many of the tools available at WuXi Pharma, and I think these are good examples of some of the technologies today. I'd like to look at the particular effect of one substitution that is commonly used in greater understanding, and that provides, I think, something of a summary and an outlook for oligonucleotides. So first, what is Tango? Tango is an acronym that describes the technology of Stoke Therapeutics. It represents targeted augmentation of nuclear gene output. And the idea is that we hope with our oligonucleotides to restore protein levels by increasing protein production from a functional copy of a gene. The idea is that this is a safe and useful approach because it selectively boosts only the gene where the protein is normally expressed. It offers one drug for diseases caused by different loss of function mutations, and it can be applied to genes of diverse sizes, whether large or small. Let me tell you a little bit about that with the following cartoon. On the left-hand side, we have a cartoon of DNA, a normal wild-type DNA. In the translation of that into the transcription of that into pre-mRNA, sometimes there is included a nonsense-mediated decay exon. Nonsense-mediated decay means that that RNA will be degraded, that there's something wrong with it. In the splicing of pre-mRNA to productive mRNA, sometimes that process will include the NMD exon. And that results in the degradation of that mRNA, as shown here, and no protein will result. Whereas what one wants is the productive excision of that nonsense-mediated exon to create a productive mRNA and therefore a protein. So the concept of Tango is to use an antisense oligonucleotide that promotes the nonsense-mediated decay exon exclusion. By targeting that oligo to a particular site on that pre-mRNA, it's possible, therefore, to generate the productive mRNA, the desired RNA, which results in a greater production of the desired protein. So we think that this is a useful approach to a variety of haploinsufficient diseases. Now, my job as a chemist is to optimize these oligonucleotides. And to do that, I often think about what are the various events that oligonucleotides will go through in this process. First, they need, after dosing, to approach some cell surface and bind to that surface. They need to get into the cell, usually by some form of endocytosis. They need to exit from the endosome, migrate to their target. They need to bind to a target mRNA or spliceosome complex. The splicing must occur and then disengage from that target and re-engage with another mRNA target. Now, I wanted to look in detail at some of the activities going on in terms of binding at the RNA target. Specifically, some relative changes in thermodynamics and kinetics and quantitative thermodynamics that are result by the substitutions, chemical substitutions that we might make. Now, as some of my colleagues or previous speakers have talked about, I think there's been a lot of progress in oligonucleotide chemistry over the years, and I'll speak about that. When the oligonucleotides come to me, they're usually in the form of phosphorothioate 2' MOE, 16 to 18 mer oligonucleotides. PS means phosphorothioate substitutions that are shown here. They're done for stabilization of oligonucleotides, as phosphodiesters are highly metabolized by exo and endonucleases. 2' MOE is a common methoxyethyl ether 2' is a common substitution also for affinity and stabilization of oligonucleotides. So what we want to do is to enhance the target mRNA engagement. We want to increase protein expression, and we want to have some understanding of the mechanism by which this oligonucleotide functions. The requirement, what I need to deliver, is an oligo having high affinity to specific sequences, that is metabolically stable, has cellular uptake, is novel or patentable, is amenable to multiple oligosynthesis, and also feasible to a CMC scale-up. Now, as mentioned, there's been a lot of progress and evolution over time in oligonucleotide chemistry, and it is not by mistake that I took a rather old reference, this by Samvi and Cook from 1994. Since that time, there have been evolutions in terms of multicyclic oligonucleotides, but I thought it was very interesting to look back and to realize that so much of oligonucleotide chemistry has been built upon a long discovery of different substitutions that one can make to backbone linkages, as well as to substitutions in stereochemistries at this 2' position and different sides of the nucleobases. So one of the substitutions, so a lot of that work has also been facilitated by the technology development. So here are shown what I call the workhorses of oligonucleotide synthesis. A Mermaid 12 is an instrument where we can make anywhere from 1 to 11 syntheses at once, or on an ECTEF system is where you can do a scale up of 1 synthesis at a time. There's also a Mermaid 192 that's by which one can make 96 oligonucleotides at a time. These are commonly used instrumentation in-house as well as at many other institutions. They use what are called phosphoramidites, which are shown here. These, again, a lot of chemistry efforts over the years have gone into creating these flexible systems that allow for very high efficiency in coupling and make possible the synthesis of long oligonucleotides. Without the framework and the basis of this chemistry, we simply could not make molecules of this size or complexity. Whereas today, we make them quite routinely in scores, if not hundreds. All right, so moving on, what's the chemistry I want to talk about today? Well, in the course of investigating the SAR exploration, we did what was called a fluoro walk. So that is a substitution of a single fluoro compound, 2' fluoro, of the same stereochemistry for the 2' MOE. So that was walked along the oligonucleotide and measured for its effect in terms of degrading the nonsense-mediated decay, as measured by an EC50 measurement. So the parent, that is the fully phosphorothioate, fully MOE compound, had an EC50 of about 5 micromolar. And you can see in this case that as we walked that fluoro compound through, we saw different levels of activity. Where the bar is higher, that oligonucleotide was somewhat more potent than the fully 2' MOE. Where that bar is lower, it's somewhat less active. So we saw that there was a positional effect of that 2' fluorination, and I became very interested in that because as we started to combine these fluorine substitutions, we saw some very different effects. So in order to investigate that, I went first to the literature. So again, drawing upon a lot of understanding and study of oligonucleotides, one can begin to wonder if it has something to do with oligonucleotide conformation. So the ribose ring, as it is substituted in DNA and RNA, is always existing in what's called a pucker conformation. It may be a C2' endo pucker, that is that the C2' is on the same side of the nucleobase and the 5' phosphodiester, or it may be a 3' endo in which that conformation is reversed and the 3' position is up, whereas the 2 is down. It is thought that 3' endo is more RNA-like, and therefore, if one is binding to RNA targets, an RNA-RNA helical type may be more stable. Both 2' MOE and 2' fluoral seem to encourage the 3' endo configuration, but since both do so, there must be more to the story to explain some of our observations. So in order to investigate that, I thought it would be interesting to do some detailed biophysical investigations here. So here, working with some scientists externally, first we were looking at what's called Biolayer interferometry. It's a form of SPR. In this case, the complementary target RNA with a biotin label is appended or is bound to a surface that is resonated with the laser in a very precise manner. Across that is washed the oligonucleotide in question that contains that 2' fluoro substitution. And then we can look at the change of the resonance of that surface with the binding of that oligonucleotide, creating this helical structure. From this, we can calculate then the stability constants as well as the dissociation and association constants. So there's a lot of information that can be derived from this technique. In a related but similar but different technique, we use the same RNA target with a biotin handle, but this time it's fully in solution, and we inject small aliquots under very highly controlled conditions of a complementary oligonucleotide, again containing that 2' fluoro substitution, and measure the heat that is released by that technique. This tells us about the thermodynamics of this association. So the hybridization releases heat, and it does so in a different way as a function of the substitutions that we found. So at this point, let me show you a little bit about what some of the results that we saw from this. So here in this cartoon, we are looking at the different numbers of fluorines that are substituted at different positions. So the parent, having a fully 2' MOE substitution, has a certain thermodynamic or a certain equilibrium constant, so 1/kd. In this case, the higher the bar, the tighter the binding interaction. So a single fluorine substitution increases the binding affinity anywhere from 26 to 38-fold in this 0.2 ppb 40 degrees. So these conditions are very important for the relative comparisons of the thermodynamic affinities. However, in simply increasing the number of fluorines does not necessarily give you a higher number. So 6 fluorines in one position or 6 in another seem to have differing results. 12 fluorine substitutions in this 18 oligonucleotide have entirely different things. So one of the conclusions we can make so far is that the site-specific nature of the 2' fluoro substitution has a different effect on the stability constant. In terms of the overall delta Gs, we see that there's not so much of a change. A single fluorine or 6 or 12 indeed enhances the delta G change, but we don't detect that as much in terms of the site-specific nature or number of fluorine counts. Now, in terms of the kinetics, we also see some interesting effects. In this case, we're looking at single fluorine substitutions, whether here at this position from the 5' end, here in these two in the middle, or this at the 3' end. In these two graphs, we can look also at the kinetics association, or here in orange, the dissociation, or again, the stability constants. So there's a lot of data on this slide. Let's start with the stability, with the association constants. When the fluorine is substituted at the 5' end, we see some enhancement of the association constant relative to the fully 2' MOE substitution. However, there's something special about these single central fluorine substitutions, a dramatic enhancement of the rate of association of the oligonucleotide, again, less so at the 3' periphery. In terms of the stability constants as well, the single 2' fluoro substitutions seem to enhance the KDs or make them lower, this is more stable, and also reduces the dissociation constants relative to the single substitutions at either end or relative to that oligonucleotide that lacks any fluorine substitutions. So we can conclude from this that the 2' fluoro substitution of central oligonucleotides has a bigger effect. So what do we conclude from this? Some things I think that are very useful. First, in a site-specific manner, single 2' fluoro substitutions have conferred enhanced target engagement on several oligonucleotides, and these have indeed resulted in increased protein expression. That is why we pursued them. We saw some advantage with these single substitutions. Single 2' fluoro substitution at central oligonucleotides result in lower KD, that is the stability constant, as well as lower dissociation constant and higher association constant, perhaps accounting for the lower target engagement EC50s as measured by the cellular pharmacology. We found, however, that multiple 2' fluoro substitutions change the activity of these oligos, at times reducing or even directing the target engagement activity. So what we see from this and have learned since is that oligonucleotide SAR is actually quite complex. It is not a simple additive thing. One is good, so two must be better. It's highly sensitive to the positions of the substitutions along that oligonucleotide chain. It is possible as well that multiple 2' fluoro substituted oligos bind to different splicing proteins compared to their PS, MOE, and or singly 2' fluoro modified oligos. Such bindings may explain the observed SAR complexities. That is data that I've not shown, but in some cases we could not even measure the target engagement for multifluorinated systems. So I hope today that I've given you some of the flavor of the details that may be necessary in order to investigate when looking at the optimization of oligonucleotides. I think it's a pleasure to speak here along with other scientists who detail some of the tools and technologies necessary in order to conduct this research. So anyway, my conclusion about next steps and how to pursue the SAR studies of any oligo is to make and assay many more oligos to understand a complex multidimensional SAR. Thank you everybody for your attention. My name is Rob Goodnow. I'm from Stoke Therapeutics. It's been a pleasure to speak to you today. Thank you.
Okay, fantastic. Well, thank you to all of our speakers for those great presentations. Well, where we have gone a little bit over, but we will just fit in some time to answer a few of your questions that have come through during the presentations. So the first question that I have is, will you also apply some direct binding assay, such as microscale thermophoresis analysis? I'm not sure if any one of our speakers would like to take that question.
Yes, we do have biophysics platforms can do such kind of assay.
Fantastic, thank you very much. Another question that we've had come through is asking about how to design siRNA or miRNA bound targeting ligands, and can you please suggest any tool for drawing these structures?
For siRNA design, there are a lot of public tools can be used, for example, OligoWalk and so on. So we have selected some public tools and also use some our own program for the sequence design, for siRNA sequence design. But for microRNA, currently we don't have the established flow chart and pipeline. Yeah.
Brilliant, thank you very much. And then another question that we've had come through is asking, what is the biggest challenge for this mode of therapeutic?
Well, this is Mark. I can probably answer that if you want. I mean, there's been some fantastic progress over recent years, particularly chemistry of the oligonucleotides, so that's increased, has led to great affinity of the oligos to different targets, also reduced the off-target effects, and that's obviously been covered to a certain extent by the speaker today. We've also now been used to treat more common diseases rather than rare diseases, which was, oligos were basically a rare disease niche market, but now they're being used for more common ailments, for example, hepatitis and hypercholesterolemia. So therefore that has extended the uses of therapeutic oligonucleotides. I suppose one area that still needs to be expanded further is to get oligos to other types of tissues, and so therefore the targeting moieties and things that we heard from Wuxi today could be used towards that. And then I guess another area is the manufacturing and cost of oligonucleotides, and that will be covered tomorrow, or whenever the next webinar is, the second webinar in this series. So, so I would suggest that I would say two things. One is we've learned a lot from Wuxi today about the progress that's been made with oligos and synthesis and design. And in the next webinar, we're going to hear a lot more about the manufacturing. So I'd recommend people dial into the second webinar.
Brilliant, thank you very much. Another question that we've had come through is saying, I may have missed it, but I assume that WuXi can assist in the design as well as the synthesis of oligonucleotides.
Yeah, yes, we can assist the design, siRNA sequence design now. And for ASO sequence design, we haven't validated the pipeline. Hopefully, we can provide the sequence design for ASO later this year. Yeah. For synthesis, I think my colleague Jun has in his presentation that has covered the synthesis part. Yeah.
Fantastic, thank you. And I think we have time for just one more question, and that is, what is the most common ASO chemistry in the pipeline and market?
I can handle that one. So I think that the phosphorothioate 2' MOE or methoxyethyl ether is probably the most common to date. One, because of the stability of the oligos in metabolic systems, but I think also just familiarity. People know a lot about it already. I think there are other chemistries that, as they're implemented in clinical experimentation, will become useful. But for the moment, these are the ones that people count on. Thank you.
Brilliant, fantastic. Well, we have just about run out of time there, but I would like to thank all of our presenters for their great presentations and insight they've shared today. And I would also like to thank our sponsor, WuXi AbTech, for making this whole event possible, as well as our follow-up webinar, which is taking place tomorrow. So on behalf of WuXi AbTech and Pharma Intelligence, we hope that you all have a great remainder of the day, and thank you very much for tuning in today.